Hookworm infection. It is an important cause for iron deficiency anemia. Hookworm are of two species, Ancylostoma and Necator americana. Ancylostoma is an old world species and Necator americana is new world species. Morphology of hookworm. It consisting of mouth, excretory pore, lateral line, anus, tail. Sex of hookworm can be distinguishable. That is, both male and female hookworm can be found. The size of hookworm is about 11 to 13 millimeter long, and L1 larva stage of hookworm is called rabidiform larva. L3 larva stage of hookworm is called filariform larva. L1 larva is called rabidiform larva. It consisting of double bulb esophagus, genital primordium, intestine, and anal pore. Hookworm species Ancylostoma and Necator can be distinguished by morphology, adult worm, and by L3 larva stage, and they are indistinguished by egg and L1 larva stage. That is, both species look alike in these conditions. Hookworm life cycle. Transmission of infective form, that is L3 larva stage, that is filariform larva, through skin penetration in human, and further these L3 infective form larva penetrate into venous circulation, and further it moves into lungs, and if the person swallow the infected sputum, then the infected sputum reaches to GIT. Further, the larva develop into male and female adult, where and further there is an fertilization going to take place, and there is a formation of an embryonated egg, and it is excreted in feces. In soil, further the life cycle proceeds, where L1 larva stage is converted or developed into L2, further L3, and the life cycle repeats as same. Lab diagnosis of hookworm infection. It mainly consisting of three stage: stool microscopy, stool culture, and molecular diagnosis. Stool microscopy. It is used to detect hookworm egg rabidiform larva, that is L1 stage of larva, and it also used in egg counting, that is number of eggs per gram of stool, and the technique used is Catho-Card's method, and the number of eggs estimate the disease burden. Then hookworm egg is of oval, 16 to 40 micrometer in diameter, and egg are covered by an eggshell, non-bile stain, colorless, saline mount, and ovum is segmented. 4 blastomere can be seen. There is a space between eggshell and embryo. They floats on saturated salt solution. Ancylostoma and Necator are indistinguishable by seeing eggs. Next diagnosis is stool culture. For this, there is a requirement of freshly passed stool to differentiate Ancylostoma and Necator by the help of L3 larva, that is filariform larva. And the culture technique used here is Arada Mori filter paper tube method, and agar plate technique, which is more sensitive culture technique. And the last lab diagnosis is molecular diagnosis, where polymerase chain reaction is involved, and it also helps in distinguish ancylostoma and Necator species of hookworm.